Hello friends, welcome for a new video tutorial. In this video tutorial, we will discuss in detail about the seed and the germination of seed. Before that, let's review the part which we discussed in the previous video tutorial, that is the development of zygote to form a mature embryo. So during the development, the zygote, which is the result of fusion of male and female pronuclei to make a diploid cell, which we call the zygote, it starts dividing. So after the division of the single cell zygote to multicellular structures, the zygote undergoes a pre-embryonic stage. So it undergoes pre-embryonic stage, which again after division it makes a globular embryo. Because it is globular, because it looks spherical in structure, then some modification occurs in the structure, making the embryo heart-shaped. So it is the heart-shaped. Then after this, a complete mature die-cut embryo is formed. Here it is represented, the mature embryo is represented with this diagram, and it is the structure of a dicotyledon mature embryo. Here these are the two cotyledons. This place is known as the plumule, the highest place of the radicle. Radicle is the mass of cell. So in this video tutorial, let's first discuss the two different types of embryo, the monocut and dicut embryo. So to understand this in detail, let's draw the diagram of the monocut embryo and dicut embryo, and let's compare these two. So friends, now we have the two different diagrams representing the embryo of the dicot and the monocut plants. So this is that of the monocut embryo and this is the dicot embryo. So let's discuss these two important types of plants and the structure of their embryo, and let's learn different parts. So first let's discuss about the dicot embryo. So the dicot embryo, the typical dicot embryo has embryonal axis along with the two cotyledons. There are two different cotyledons, that's why it is called dicot embryo or dicotyledon plants. Okay, so the cotyledon is here 2 in number. Next, plumule. So the plumule is this is the hypocotyle, the mass of cells present in this region, and above the hypocotyle, the end part from where the two cotyledons divide into two different parts. This highest position of the hypocotyle is the plumule. Okay, at the base we have the radicle, and covering the radicle there is the root cap. Okay, so the root tip is covered with a root cap, okay, for protection. So the cylindrical part below the level of cotyledon, is the hypocotyle. This is the cylindrical part which is present below the level of the two cotyledons. Okay, so hypocotyle terminates at its lower end in the radicle. The hypocotyle terminates at the radicle. Okay, or the root tip. It is also known as the root tip. And that root tip is the lowermost part of the hypocotyle, which is also called radicle, and the root tip is covered with the root cap. Okay, so the root cap is present at the base. Next, let's move to the monocut embryo. How the monocut embryo looks like and what are the different parts. So here the highest part is the scrutellum. What is scrutellum? It is nothing but the cotyledon. So the cotyledon in case of monocut embryo is known as the scutellum. Okay, it is, we are talking of the grass family. Cotyledon is called scutellum. Remember, this is nothing but the cotyledon, which is called scutellum in case of monocut embryo. So it is situated towards one side of the embryonal axis. If this is the center, it is present at one side, it is not present at the middle. So the cotyledon is laterally located or positioned laterally. Part of embryonal axis above level of attachment of scutellum is epicotyle. It is the epicotyle which is located, it is the part of embryonal axis above the level of scutellum. Epicotyle has a shoot apex and few leaf primordial enclosed in hollow foliar structure. Enclosed in a hollow foliar structure which is known as coleoptile. So you can see here the coleoptile is present in this region. This is the coleoptile which covers few leaf primordial. So here the some leaf-like structures present, which is enclosed with the help of the coleoptile. Okay, the shoot apex is also located here. We can see this shoot apex. So shoot apex, the epicotyle has a shoot apex and few leaf-like structures which are enclosed in a structure called the coleoptile. It is called coleoptile. Okay, so this is all about the two different types of embryos, the monocut embryo and the dicut embryo. In case of monocut embryo, one more thing we can notice from the diagram. Here the radicular root tip is the last part of the hypocotyle, which is covered by the root cap. But here the radicle is covered by the root cap and the root cap is also covered by another structure here, coleorhiza. So this is the difference between these two types of embryos, the monocut and dicut embryos. So this is the dicut embryo that I have discussed in the previous class, and this is the mature dicut embryo, and its modified form is like this. So this part, the hypocotyle is here, this one. The radicle is at the base of the hypocotyle. This is just inverted here. So plumule is present here, here plumule is present here, and these are the two cotyledons, here these are the two cotyledons. So nothing to puzzle between these two, and this is the suspensor from which this structure is modified from the heart-shaped embryo during the developmental stage. You can see here from zygote to heart-shaped embryo, and then only mature embryo is formed. So the heart-shaped embryo is modified more so that the cotyledons can be easily seen in this case. So after modification we can make the diagram like this. This is the perfect mature dicut embryo. Okay, so this is all about the structure of embryo, the development of embryo in case of monocut, how the embryo looks, in case of dicut, how it looks, how zygote develops into a mature embryo. Okay, so now we will move on to the structure of seed and its different properties. So let's discuss about seed now. So the seed is formed after the modification of ovule. The ovule is modified to form the seed, and the ovary is modified to form the fruit. So after fertilization all these occurs, so this also comes under the post-fertilization events. So let's discuss about the seed. Okay, so seed is the final product of sexual reproduction. It is the final product. Finally, we will get the finally we will get the seed. It is the final product of the sexual reproduction in plants. Sexual reproduction in plants. So it is the fertilized ovule. As I have told you, it is the modified ovule. So ovule when fertilized makes the seed. So the fertilized ovule is seed, and it consists of seed coat as its outer covering, then cotyledons when 2 it is dicot, when 1 it is monocot, and finally the embryo axis or embryonal axis. Okay, so these are the 3 important parts of the seeds. So what is cotyledon? Let's learn. So cotyledon is the thick swollen part. It is a thick and swollen. Why it is thick and swollen? Because much more amount of nutrients are deposited here due to nutrient deposition. Okay, so nutrients are deposited here, or the storage of food. We can also call it storage of food. Due to storage of food in the cotyledon, it looks thick and it is swollen. So mature seed may be non-albuminous, or albuminous. So it may be albuminous or non-albuminous. So when it is non-albuminous, seeds have no endosperm, no residual endosperm. So endosperm provides nutrients for the developing embryo. We know that when the zygote develops into embryo, during its development, the endosperm provides the necessary nutrients, or it stores the food material that is utilized during the development of the zygote. So when all the nutrients that is stored by the endosperm is utilized, no residual endosperm is there, at that time it is called non-ambiguous seed. The completely consumed, which the endosperm is, in this case the endosperm is completely consumed by the embryo, completely consumed by the growing embryo or developing embryo. Example, we can take the pea and groundnut. Okay, pea and groundnut are the example of non-albuminous plants. Next, let's move on to the albuminous plants or albuminous seeds. They retain part of endosperm. They retain some part of the endosperm. So when the endosperm is not fully consumed or not completely consumed during embryo development, it is called albuminous seed, and they retain part of the endosperm. And the examples can be the wheat, maize, barley, sunflower, etc. Wheat, barley, sunflower, etc. These are the examples of the albuminous plants. Non-albuminous are the pea and groundnut. In case of albuminous, where the part of endosperm is written by the embryo or not fully utilized, in that case it is called albuminous seeds, and the examples are wheat, barley, sunflower, maize, etc. Okay, so let's move on to the special cases. In some seeds like the black pepper and beet, the nucellus is persistent. The nucellus is the outer covering of the ovule. Nucellus is persistent. Nucellus is persistent in case of black pepper and beet, and it is residual persistent nucellus. The residual persistent nucellus is called perisperm. It is called perisperm. So nucellus, what is the function of the nucellus in the ovule? The ovule is surrounded by the nucellus, the cells that contains the necessary nutrients for the development of the ovule. So that is the nucellus, which is again covered by the integuments, two different parts. Integument has the inner integument and the outer integument. Okay, at the center ovule is there. So nucellus provides the necessary nutrient material to the ovule, and here the nucellus is persistent, and when it is residually persistent, the nucellus is called the perisperm. As the endosperm, here we know endosperm provides nutrients for the growing zygote to form the well developed or mature embryo. Endosperm provides the nutrients, but here the nucellus provides the nutrients for the growing of the embryo. That is the inside the ovary, the ovule grows with the help of nucellus, which is known as the perisperm, or the persistent nucellus is called the perisperm, which provides nutrients for the growing embryo, ovule. Okay, so the ovule develops with the help of the nucellus, and the integument, which covers the nucellus, works as the seed coat. As the ovule develop into seed, ovule when fertilized develop into seed, and the integument that covers the ovule now becomes the seed coat. As in the case of unfertilized ovule, when the ovule is not fertilized, the ovule is surrounded by the integuments, and after fertilization the ovule becomes seed, and the integument changes to seed coat. So the seed coat is formed from the integument. And another thing we know that the micropylar end is the one through which the pollen tube enters into the ovule so that it can reach the ovule. So the micropylar end is also present here, and it is a small pore. It is a small pore in between the integuments. As we know, in the structure of the ovule, the integuments present throughout the ovule except the micropylar end or the micropylar pore. That pore is also present after fertilization, and here the micropylar end is a small pore in the seed coat. It is a small pore in the seed coat. The seed coat is present everywhere surrounding the seed except a small pore which is known as the micropylar end. So it facilitates entry of oxygen and water. It is important, micropylar end, because it helps in the entry of water and oxygen. Water and oxygen can easily enter the seed, not to the seed coat, but to the seed. How? With the help of the micropylar end, which is a small pore in the seed coat, and through this micropylar end, water and oxygen enters the seed during germination. During germination. So during germination, what will happen? The seed or the fertilized ovule gets oxygen and water, which is very essential for the germination of seed, through this micropylar end, which is a small pore in the seed coat. So now it can easily get the essential water and oxygen for its germination. Next. So here we have discussed about what? About the nucellus, which when persistent it is called perisperm, and we have discussed about the non-albuminous and albuminous plants. Non-albuminous are where no residual endosperm is remained, so that all the endosperm or the nutrients are completely consumed during the development of the embryo. But here in case of albuminous plants or the albuminous seed, the part of endosperm is retained, which is not completely consumed during the process of development. And the example of the non-albuminous plants are the pea and groundnut. Pea and groundnut are the example of the non-albuminous seeds, and in case of the albuminous seeds, the examples are the wheat, maize, barley and sunflower. So now let's discuss about the detail of how the germination of seed takes place. So here we know that the micropylar end plays important role because it is the pore in the seed coat, so that through this the essential water and oxygen enters to the seed for its germination. So what happens when seed matures? When seed matures, its water content is reduced. Water content is reduced. So the moisture content of the seed is reduced. Okay, so it loses water and becomes dry, relatively dry, and the moisture content becomes 10 to 15% less. 10 to 15% less, and the seed becomes relatively dry by losing water. So the general metabolic activities of embryo now slows down. General metabolic activities slow down. It will slow down. Why? Because it enters a state of inactivity which is known as dormancy. It is a state of inactivity. It is a state of inactivity which is called the dormancy. So during dormancy period, the water content of the seed is reduced. It loses water, becomes dry by 10 to 15%, and the general metabolic activities slow down, because if the metabolic activities will be more, then it will require more amount of energy, and to avoid that supply, if we will not consume more energy, then it can easily enter to a stage of dormancy, which is called the state of inactivity, where the metabolic activities become slow down, and it can even become live in that condition for a longer duration of time without much nutrients. Okay, when favorable condition comes, so this will happen during unfavorable condition. When favorable condition comes, favorable condition. What do you mean by favorable condition? Here in case of the seed, for the seed, it is the good amount of moisture, sufficient amount of oxygen and good temperature, or adequate temperature, oxygen and moisture. Okay, so during this favorable condition, when favorable condition comes, what will happen? The germination of seed. Germination of seed. The seed germinates when the favorable condition appears. That means favorable amount of moisture, oxygen and good temperature or adequate amount of temperature comes. At that time, the seed from the inactive state or from the phase of dormant seed will come out and germination of seed takes place. So after germination, what happens? The ovule develop into seed, and the ovary develops into fruit. Okay, so development occurs, and these two processes, the conversion of ovule into seed and ovary into fruit, occurs simultaneously. Okay, so both the process goes together. Okay, so the development occurs together, and the ovary wall makes the wall of the fruit. The wall of ovary, as the ovary becomes fruit, so the ovary wall of ovary becomes wall of fruit, and this wall of fruit is called the pericarp. This is called the pericarp. So the wall of ovary develops into wall of fruit, which is known as pericarp. Okay, so fruits have their unique mechanism of seed dispersal. In different types of fruits, there is their unique process of dispersal of the seed. So why they disperse the seeds? In order to get different habitat in different areas, the dispersal occurs. So fruit develops from the ovary. Other floral parts degenerate. Only the ovary becomes fruits after fertilization, and the other floral parts like the petals, sepals, the androecium, the male reproductive part, all degenerates and falls off. Okay, so in some other types of fruits, we will discuss about two different types of fruits now. So I was talking about the dispersion of the seed. So in different plants, their own rule is there, their own principle is there. They always try to disperse to a variety of areas, to a wide range and to different habitat. In few species, we will discuss about some special case. So that we will discuss about few species. In few species, what will happen? The thalamus plays important role in the formation of fruit. In that case, the fruit is called false fruit, because the fruit is not only developed from the ovary, but the thalamus has certain role for the development of the fruit. Example is apple. Then we can also say strawberry, and cashew. These are the example of the false fruit. And another thing, fruit develop only from ovary. When they only develop from ovary, so ovary get converted into fruit. in that case we call it the true fruit. It is the true fruit. Okay, so in most of the plants we get the ovary converted into fruit, means most of the fruits are true fruits. Others, such as where the thalamus helps in the formation of the fruit along with the ovary, at that time it is called the false fruit, and the examples are apple, strawberry and cashew. So next, the last point that I want to discuss here with you, which is very important also. This is also very important, true and false fruit and examples. So another important is fruit that develop without fertilization. Without fertilization, fruits develop. Like in case of some animals, there is a process known as parthenogenesis, where the female, without the help of the male, the egg of female grows to offspring. Okay, in that case, without fertilization, the offspring or new individual is produced in that animal, and it is known as parthenogenesis. But here, when the fruit develops without fertilization, we call it parthenocarpy. Parthenocarpy. So do not forget this. This is very important. It is known as parthenocarpy, and the fruit is called parthenocarpic fruit. The fruit is here the parthenocarpic fruit because it developed as a result of parthenocarpy, which means development of fruit without fertilization. Okay, so here who plays important role for the process of fertilization? It is the growth hormone. It is the growth hormone. So growth hormone makes specific changes by which the fruit is developed without fertilization, and the fruit formed is known as parthenocarpic, and the process is called parthenocarpy. So here the fruits which are formed are seedless. So seedless fruits are formed, and the example is banana. So in case of banana, the fruit, that is banana, which we consume, most of us consume this banana. It is a very easily available fruit in our area, and this banana is the example of a parthenocarpic fruit, and it is seedless. Okay, so this is all about the different events that occur after fertilization. And here we learned about seed, how the seed is formed, when the seed will germinate, when it will enter to a stage of dormancy or the stage of inactivity, what is the micropylar end, what is its role, how the seed coat covers, the integument develop into seed coat, and it covers the seed except that small pore which we know as the micropylar end, through which the essential oxygen and water enters into the seed during germination. And we also discussed about the true and false fruit. Before that, we have discussed about the embryo in case of dicot and monocot plants, and finally we discussed the process of parthenocarpy and how parthenocarpic fruits are produced without fertilization as a result of the hormonal activities, and in this process seedless fruits are formed, and the example is banana. So this is all about the different events of the post fertilization changes in the plants. So in the next class we will go to the last part of this chapter and we'll finish it. And if you have any doubts in this video tutorial, then you can comment below so that I will clear you and clarify you. If you like this video tutorial, please hit the like button. It encourages me for making more videos for you. If you have not subscribed yet, please subscribe this channel and hit the bell icon. Thank you very much.